Implantation begins at 7th day after fertilization, and in the implantation the blastocyst is eroding the endometrial stroma with the help of histolytic activity of the trophoblast. So the embryonic pole of the trophoblast will start eroding in the endometrial stroma. This endometrial stroma is then divided into the three parts: stratum basal, stratum spongiosum, and stratum compactum. So the stratum compactum and spongiosum is now occupied by the developing embryo. Okay, so these two parts of the endometrium are now occupied by the embryo, and the implantation is completed, and then the site of penetration is closed by fibrin plug. Soon after implantation, this decidua is divided into three parts. So the decidua which is facing towards the embryonic pole, this part is known as decidua basalis. Then the decidua which is covering rest of the embryo, that is embryonic pole, it is known as decidua capsularis, and rest of the decidua covering the uterine lining is called decidua parietalis. Okay, so there are three parts of decidua. Facing towards the embryonic pole of the trophoblast is the decidua basalis. So this is decidua basalis. Then the embryonic pole, it is covered by decidua capsularis, and rest of the uterine cavity is lined by decidua parietalis. Similarly, the chorion is also divided in two parts. Chorion means trophoblast. So the trophoblast which is facing towards the decidua basalis forms chorionic frondosum. So this is chorion frondosum facing towards the decidua basalis, and rest of the chorion, you can see this dark brown color. So rest of the chorion is called chorionic levy. So there is interstitial implantation, and the blastocyst is embedded in the uterine endometrium. As the fetus grows in size, the uterine cavity will go on obliterating. Now you can see the developing fetus, and here there's formation of placenta. So which are the two constituents which form the placenta? It is the decidua basalis from the maternal side and chorion frondosum from the fetal side. So these two will help in the formation of placenta. At the second week of development, the trophoblast differentiates into two layers. So the inner layer of the trophoblast is known as cytotrophoblast. Here the cells are mononucleated, they have distinct cell membrane, and they are pale staining. So the inner layer is known as cytotrophoblast, whereas the outer layer is known as syncytiotrophoblast. The syncytiotrophoblast does not have limiting cell membrane. It is multinucleated and it is darkly stained. Soon after the differentiation of trophoblast into inner cytotrophoblast and outer syncytiotrophoblast, there is appearance of lacunae. Small cavities will develop, they will coalesce, and they will form such lacunae in the syncytiotrophoblast. Okay, so in the syncytiotrophoblast we can see such lacunae. So these are the empty spaces or the lacunae. When the blastocyst is eroding the endometrium, the maternal blood vessels will rupture, and the blood from the spiral vessel or spiral arteries will enter into these lacunae, and there is establishment of uteroplacental circulation. So in these lacunae there is maternal blood. So these are now known as intervillous spaces. Okay, the lacunae containing maternal blood are now known as intervillous spaces, and in between these intervillous spaces, now you can see this is one intervillous space and this is another, and in between them you can see trabeculae. So trabeculae is the part where still the syncytium is present. The syncytiotrophoblast is still present at the trabeculae. Okay. Now the cytotrophoblast will start embedding, or it will start invading the syncytiotrophoblast. trophoblast. The same can be seen here. So the cytotrophoblast is invading inside the syncytial trophoblast at the area of trabeculae, and now this trabeculae is converted into primary stem villi. So what is primary stem villi? It is the invasion of the cytotrophoblast into the syncytiotrophoblast. Okay, so the cytotrophoblast is invading into the syncytiotrophoblast at the region of the trabecules, and it is forming primary stem villi. The same can be seen here. Okay, so this is the cytotrophoblast. It is the cytotrophoblast, and this green color is the syncytiotrophoblast. Now what will happen next is the cytotrophoblast, which is invading the syncytiotrophoblast, will reach up to the tip of the primary stem villi, and now it will start covering the syncytial trophoblast, and it will entirely cover the blastocyst or the developing embryo. It will entirely cover the developing embryo at the end of third week. So this is known as cytotrophoblastic shell. Okay, so this is the final picture of the developing embryo at the end of second week. So we will see what happens first. You can see this is the cytotrophoblast. Okay, this entire is the cytotrophoblast. Then this one is the syncytiotrophoblast. These are the intervillous spaces filled with maternal blood. In between the intervillous spaces, you can see the primary stem villi, which has cytotrophoblast, and the green color is the syncytiotrophoblast. So inner cytotrophoblast, outer syncytiotrophoblast, and finally there is formation of cytotrophoblastic shell. Okay, so the cytotrophoblast or this shell will divide the syncytium into two parts. There is inner layer and outer layer. Later on, both these layers will undergo fibrinoid degeneration. So these are the changes in trophoblast occurring in the second week. To summarize it shortly, what happens first? There is differentiation into inner cytotrophoblast, outer syncytiotrophoblast. Then lacunar spaces are occupied by maternal blood to convert it into intervillous spaces. The trabecules are invaded by cytotrophoblast to convert it into primary stem villi. As the maternal blood enters the lacunae, uteroplacental circulation is established, and lastly, the cytotrophoblastic shell is formed. During second week of development, we have seen the changes are occurring in the trophoblast. Simultaneously, changes are occurring in the embryoblast also. So what changes occur in the embryoblast? The inner cell mass will form an upper lamella of tall columnar cells, which is known as epiblast or primitive ectoderm, and towards the luminal surface cuboidal cell layer is formed, which is known as hypoblast or primitive endoderm. Okay, so what changes occurring in embryoblast? There is formation of bilamellar germ disc. Towards the trophoblastic surface we have tall columnar cells, which is known as primitive ectoderm or epiblast, and towards the luminal surface we have the cuboidal cells, which is known as hypoblast or primitive endoderm, and in between these two germ layer we have extracellular basement membrane. The embryoblast will soon detach itself from the trophoblast, and a cavity is seen in between the trophoblast, the embryonic pole of the trophoblast, and the bilaminar germ disc. So this cavity is known as amniotic cavity. So the roof of amniotic cavity is formed by amniogenic cells derived from cytotrophoblast, and the floor is formed by the primitive ectoderm or the epiblast. Okay. After that, the mesothelial cells, they are derived from the cytotrophoblast, and they will line the inner aspect of the cytotrophoblast. It is known as Users membrane.
So this user's membrane will line the inner aspect of the embryonic pole of the trophoblast, and now other cavities form which is known as primitive yolk sac. So this primitive yolk sac has roof formed by the endodermal cells or the cuboidal cell, and it is lined by User's membrane which is formed by flat mesothelial cells derived from cytotrophoblast. So far we have seen bilamellar germ disc is formed and there is formation of two cavities. Okay. After that, we can see the formation of extra embryonic mesoderm. So this brown color is the extra embryonic mesoderm. Again, it is derived from proliferation of cytotrophoblastic cells. It is called extra embryonic because it is lying outside the bilamellar germ disc. And small cavities will appear in this extra embryonic mesoderm. The cavities will fuse and they'll coalesce and they'll form a large horseshoe shaped cavity in this extra embryonic mesoderm. So it is, this cavity is known as extra embryonic coelom, and it will divide the extra embryonic mesoderm into two parts. So the splanchnopleuric extra embryonic mesoderm is one which is lining the yolk sac. So this is the splanchnopleuric extra embryonic mesoderm lining the yolk sac, and rest of the extra embryonic mesoderm which is lining the amniotic cavity and the inner aspect of the cytotrophoblast is known as somatopleuric mesoderm. So far we have seen formation of bilamellar germ disc, formation of two cavities, formation of extra embryonic mesoderm, extra embryonic coelom, and division of extra embryonic mesoderm into two parts, that is splanchnopleuric and somatopleuric mesoderm. Now this coelomic cavity is extending in horseshoe shaped manner. Here also you can see this is the extra embryonic mesoderm, sorry, coelom. So this cavity is known as extra embryonic coelom, and it is attached only at one point. So the attachment of the developing embryo with the trophoblast at the caudal part by the extraembryonic mesoderm is known as connecting stalk, which will form in future the umbilical cord. Okay. So this is the connecting stalk which is connecting developing embryo with the trophoblast. After this change, now what next change occurs is the yolk sac will reduce in size. The size of the yolk sac, primitive yolk sac, will reduce, and large portion of it is pinched off by the traction applied by extra embryonic mesoderm. So now the yolk sac is left as a small cavity, and now it is lined by the cells derived from endoderm. So endodermal cells will line the yolk sac here, and this yolk sac is now known as definitive yolk sac. So initially we had primary yolk sac which was large in size, and the cells or the user's membrane, the cells were derived from the cytotrophoblast, whereas in the definitive yolk sac, the size is small and it is lined by the cells which are derived from the endoderm. Now after this change, the next change occurring is formation of precaudal plate. So we have seen that the towards the outer surface or towards the trophoblast we have epiblast, and towards the luminal part we have hypoblast or endoderm. Now the endodermal cells towards the cephalic part, they will differentiate from cuboidal to tall columnar. Okay, so there is differentiation of cephalic cells of the endoderm into tall columnar cells which are firmly adherent with the ectodermal cells. So this precaudal plate is formed at the cephalic end, and it is the first sign of differentiation of the germ cell. And there is again formation of cephalocaudal axis. So we can say in the germ layer, the part where the precaudal plate is present is the cephalic part, and the part where you get the connecting stalk is the caudal part. So you have the precaudal plate in the cephalic part, and towards the caudal part you have the connecting stalk. Also, bilateral symmetry of the embryo is established by the formation of precaudal plate. So these are the changes occurring in the second week in developing embryo. So we will revise the changes occurring in trophoblast and embryoblast at second week of development. It is known as week of twos. It follows rule of two. Okay, here the trophoblast is differentiated into two layers: inner cytotrophoblast, outer syncytiotrophoblast. Embryoblast is differentiated into bilamellar germ disc with upper lamella of epiblast and lower lamella of hypoblast. Then two cavities are seen. There is amniotic cavity and yolk sac. The yolk sac is again of two types. Initially we have primitive yolk sac. It is then converted to secondary or definitive yolk sac. Then two fetal membranes are there, amnion and chorion. So chorion is the membrane formed by the trophoblast and the somatopleuric extraembryonic mesoderm. It will form the chorion. Whereas amnion is the membrane formed by amniogenic cells of the amniotic cavity and the somatopleuric extraembryonic mesoderm. Then there is formation of extra embryonic mesoderm between the trophoblast and these two cavities, and soon after that there is appearance of a horseshoe shaped coelom in the extra embryonic mesoderm, which will divide it into two parts. So the extra embryonic mesoderm is divided into splanchnopluric and somatopleuric mesoderm. Then two cavity membranes are there, amniogenic lining the roof of amniotic cavity, and initially in the primitive yolk sac we have the Users membrane. Then two ends of the embryo are cephalic end and caudal end. So towards the caudal end we have the connecting stalk, towards the cephalic end we have the precaudal plate. And two types of villi. Okay, we have mentioned about the villus also in the trophoblast. So primary villus is the one in which the inner core is formed by the cytotrophoblast and the outer core is formed by syncytiotrophoblast. And after the formation of extra embryonic mesoderm, the extra embryonic mesoderm will also invade in the villi. So we have the innermost core formed by extraembryonic mesoderm. Then there is a layer of the cytotrophoblast and outer layer is of syncytiotrophoblast in the second. Now we will see some interesting aspects of the clinical side or the clinical significance of second week of development. So we know that the implantation occurs in which the blastocyst is implanted in uterine cavity. Now this blastocyst may act as a foreign body or an antigen to the mother. Okay, means it is a form of allograft, we can say. What is allograft? Transfer of tissue from a member of one species to another member of same species. So here there is transfer of fetus into the mother. It is human to human transfer, so it is also known as allograft. Now normally when a graft is there or when a foreign particle enters into our body, we treat it as antigen and immune response will occur. There is formation of T cells and they will act or they'll attack on the antigen. But this does not occur during pregnancy. The mother accepts the conceptus or the fetus without inducing any immune reaction. So how it is possible? The immune reaction, how it is generated? Now the antigen, for example here the fetus or the conceptus, it will bind with a major histocompatibility complex molecule which may induce immune reaction on exposure to the mothers, and it may cause differentiation of T cells, but such response does not occur. And what are the factors or the reasons be, uh, because of which the fetus is accepted and there is no induction of immune reaction or there is no rejection of the fetus? So the important factors are: first is physical separation. The trophoblast will separate the fetus from mother, and this trophoblast has poor expression of major histocompatibility complex which is responsible for immune reaction. Then in the mother, specific hormones are released. They will cause maternal immune system to become non-responsive to the fetal tissue. Then third is the conceptus has immature, means it is antigenically immature. Then the trophoblast from the fetal side will also produce tumor necrotic factors which will induce apoptosis in the activated T cells of the maternal side. So these are all factors due to which the fetus is accepted and there is no rejection of the fetus. And now the second interesting clinical part that we can see here is the genomic imprinting. So what is this genomic imprinting? It is the phenomenon that involves expression of genes depending upon its maternal or paternal origin. So there will be different expression or different clinical scenario depending on the same gene from either maternal side or paternal side. So we'll see how it occurs. So for example, now the zygote, normally the zygote contains one male pronucleus and one female pronucleus. Okay, normally, and there is fusion of the male pronucleus with the female pronucleus to form a zygote. But if there are two male pronucleus and the female pronucleus is absent, in such case the trophoblastic mass is formed. Okay, when there are two male pronucleus, trophoblastic mass is formed. It is known as hydatid form mole and it may lead to malignant change called choriocarcinoma. Now the other scenario is if the zygote is formed by two female pronucleus, and here if the male pronucleus is absent or if it is inactive, then it will give rise to non-surviving embryoblast, and it will also lead to malignant change at times known as ovarian teratoma. Okay, so the same zygote is there, but if there are two male pronuclei, it will lead to formation of trophoblastic mass which may form hydatid form hole, and if the zygote is formed by two female pronuclei, it will give rise to embryoblast which will not survive. Okay, then the second, now what is why this occurs? It is due to genomic imprinting. So the DNA of female germline is more methylated than the DNA of the male germline, and this is the basis of genomic imprinting. Methylation of DNA leads to either gene silencing or gene activation. Now other example is if there is deletion of long arm of chromosome number 15 in case of males, means if this chromosome is from the father side, it will result in Prader-Willi syndrome, and if the chromosome is from the maternal side, it will lead to Angelman syndrome. Okay, then the next is the Barr body. So what is Barr body? In females we have one inactive X chromosome. We know that in the female embryoblast there is a pair of X chromosome, means there are two X chromosomes, so one of it is inactive irrespective of its parental source, may it be from maternal side or paternal side, but one X chromosome is always inactive. And in the trophoblast of female blastocyst, the paternal X chromosome is inactive. Okay, in the embryoblast, the inactive X chromosome can be either maternal or paternal, but in the trophoblast of female blastocyst, the paternal X chromosome is always inactivated, and the inactivation of X chromosome involves expression of specific X chromosome locus. It is known as X inactive specific transcript gene, and it causes methylation of X chromosome. And after methylation, in the methylated part, there is lack of histone H4 acetylation, and this results in condensation of X chromosome and it will form the Barr body. So inactive X chromosome can be reactivated in early fetal life, but then it remains dormant throughout the remaining life. Okay, so the inactive condensed X chromosome is known as Barr body.